Chapter 16, question 9 asks, what type of interaction would you expect between the R groups of the following amino acids in a tertiary structure? So, we talked about in the Chapter 16, question 8, we talked about the 4 different levels of structure for a protein. Primary structure is the order of amino acids. Secondary structure is local wrinkling in an amino acid chain. Tertiary structure is the global folding of a chain. And quaternary structure is when 2 or more chains clump up without bonding to each other. So here, they're asking us about this tertiary structure. Let's talk for a second about how that forms. So, for most of these proteins, they're going to be, on Earth anyway, they're going to be in an aqueous solution. That is a solution of water. So these proteins are going to be completely surrounded by water. Now, we saw in table 16.2 how some amino acids, the R groups on them, are polar, and some of them are nonpolar. Okay. Well, what happens if you have a chain of amino acids? So here's our amino acid chain. And some of the R groups are nonpolar. And some of the amino acids are polar. Well, the nonpolar molecules will not want to be surrounded by water. They do not mix. Like dissolves like, water is polar, and these R groups are nonpolar, so they are not happy. They're very uncomfortable. On the other hand, the polar R groups are very comfortable. They are polar and water is polar, so they don't mind being surrounded by water. So what ends up happening is the protein folds. So here just a very simple fold. The protein folds so that all the nonpolar amino acids, the amino acids with the nonpolar chains, are with each other in the center of a glob. And the polar amino acids are outside of that glob, facing the water. And that is how these amino acids, the nature of the R group, can determine the 3-dimensional shape of a protein when it folds. So, these interactions between the nonpolar R groups are called hydrophobic interactions. That's between nonpolar R groups. Remember, the R group is that 4th group in an amino acid that determines which amino acid it is. Those are the hydrophobic interactions. On the other hand, you might have a couple amino acids, R groups, that really want to be near each other, and so, because they're both polar. So, if those amino acids come together because of their polarity, then that attraction between them would be hydrophilic interactions. The attraction between 2 polar R groups are hydrophilic interactions. So you can see how that concept that we first ran into in terms of solubility, like dissolves like, is profoundly important for how proteins fold and therefore what they can do. The nonpolar R groups will go with other nonpolar R groups. The polar R groups will go with other polar R groups and face the water. And that determines the shape of the global shape of the protein. The global shape is the tertiary structure. There's one other, aside from the hydrophobic interactions and hydrophilic interactions, there's one other type of interaction that can be really important in terms of creating the global shape of a protein. In other words, in terms of creating its tertiary structure. And that is what's called a disulfide bond. If you go back to table 16.2, that lists the 20 most common amino acids in your body, there is one of these, cysteine, that plays a profound role. If you had two cysteines, notice at the edge of cysteine you have that SH. If you had two cysteines, and I won't draw the whole thing, but you'd have another blue box on top there. If you had that SH and you had two of them facing each other, these can snap together and you would have, so this other part is not of importance right now. This rest of it, not of importance right now. They would snap together and form a bond. And because that bond has two sulfurs, this bond right here, because that bond has two sulfurs, it's called a disulfide bond. And it is a covalent bond. Notice that the other two things that formed tertiary structure were hydrophobic interactions and hydrophilic interactions. That has to do with things that aren't bonds. They're weaker than bonds, and so you can easily unfold these proteins. Disulfide bonds are full covalent bonds, so it's very, very hard to undo them. Just to give you an example of what that would look like, let's say you had a protein again, what the influence of disulfide bond would do. Let's say you have a protein, you have one disulfide bond, or you have one cysteine in one place and another cysteine in another place. Well, those can come together and form a disulfide bond. That's a really, really strong bond. That protein is going to be stuck folded that way, even in very extreme changes in conditions. So, the strength of this bond makes the protein harder. And you can sort of gauge how many disulfide bonds there are in a protein in a very qualitative way by just seeing how hard it is. For example, there are more disulfide bonds in your fingernails than there are in your hair. It's the disulfide bonds, this very rigid bond, that's making your fingernails harder. There are more disulfide bonds in a horse's hoof than there are in your fingernail. And it's the extra amount of disulfide bonds that makes the horse's hoof harder than your fingernail. Interestingly, there are also a lot of disulfide bonds in creatures that live in extreme conditions. There are some microbes that can live in boiling water, and the reason they can live that way is because no matter how violent the water molecules crash and smash into their proteins don't change shape because very strong disulfide bonds are holding them in a very certain shape. If the same hot water started to crash into our proteins, they would make them unfold because our proteins are not held together by as many disulfide bonds. There are also proteins that tend to clump together and cause diseases. One of them is amyloid beta, causes Alzheimer's disease, or is implicated in Alzheimer's disease. And in some species have evolved disulfide bonds to make sure that those proteins can never unfold or misfold to cause the disease. So disulfide bonds are profoundly important interactions that determine the global folding of an amino acid chain. In other words, its tertiary structure. So, in terms of solving question 9, we're going to look for two things. First, if we have a cysteine, if we have two cysteines, then we're going to know that we can form a disulfide bond, that very special bond. And in this first one, we see that there are two cysteines. And so cysteine and cysteine, and so the interaction that's going to hold the R groups on these two amino acids together is a disulfide bond. Okay, how about with aspartic acid and lysine? Aspartic acid and lysine, well, if we go to table 16 and we look for those two, we will see that aspartic acid, here in the bottom left, is polar. Remember, everything from the middle down is polar. Aspartic acid is polar, okay, and lysine, that is on this table in the bottom right, that's also polar. These are two polar R groups. These two R groups are polar. You can see it because they have nitrogens and oxygens in them. And so they are going to interact through hydrophilic interactions. So we can write hydrophilic interactions. Next, they have serine and aspartic acid. So we'll go down to our table, look where those are. Serine and aspartic acid. Well, serine is right here, very clearly a polar amino acid. Aspartic acid, bottom left, is another very polar amino acid. So, because we're dealing with an interaction between polar R groups, this is again going to be a hydrophilic interaction. Hydrophilic interactions are going to make those stick together and determine how a protein folds. Last but not least, D has leucine and leucine. So here, leucine is in the top right of this table, and that is a nonpolar amino acid. This R group doesn't have any partial charges. It's nonpolar. So the two leucines are nonpolar. If you have interactions between nonpolar R groups, those are hydrophobic interactions, and so these two leucines are going to interact through hydrophobic interactions. So those are the interactions that we determine the three-dimensional structure of the whole protein. If you want to visualize how beautiful these are, you can go to what's called the protein data bank. So if you type into Google protein data bank, this top one that comes up is the protein data bank, and you can search for any protein. So for example, we could search for the protein that detoxifies the alcohol you drink, alcohol dehydrogenase. Search for it, and you can look at the three-dimensional structure of the protein for free. Now, here we have it, the three-dimensional structure, and in this structure, we can have a 3D view. You can move it around, see the three-dimensional structure, the tertiary structure, and in this tertiary structure you can see the secondary structures. Notice the spiral staircases or the springs, like this red one here on the left. Those are alpha helices. And sometimes you'll have like a sort of straight ribbon, like on the left here in the light blue. That's a beta pleated sheet. So you can see that secondary structure in here. And if you wanted to, at the protein in the protein data bank, you can also get different views, including views of involving individual atoms. And so this is how we've been seeing elements through the organic chemistry, where the gray is carbon, white is hydrogen, red is oxygen, blue is nitrogen, here you can see how all of those combine to give you that three-dimensional tertiary structure. And you can find this for any protein almost in the human body for free.